welcome back to the lecture series, the application of nanomaterials in agriculture. So, now we have moved into the section where we are talking about the application of nanotechnology in animal production. So, if you recollect back, we talked about some of these areas, what we enumerated in the dairy sector for milk from the cows and buffaloes, camel, goat and sheep. These needs to, in order to feed, you need to increase the milk production. And when we talk about the milk production, essentially formation of milk from the blood, when it happens in the mammary gland of the female part of the species, it consumes a lot of macro and micronutrients, and milking mother needs to be supplied with these different micronutrients in sufficient amount, and only way you can supplement it is through food. And not only in milk production, in terms of the body weight, in terms of the meat production, that's equally important. So, if we talk about the other sector, which is the meat, which includes your poultry, beef, goat and several other animals, then we talk the fisheries, which is of two sections. One is the fish as a food, the other one is the ornamental, and then the insect sector we talk about lac, silkworm and their commercial significance. The areas where major chunk of investments are happening is this area: food and food additives, nutraceuticals for the animal production. Okay, this is for animal production, because nutraceuticals could be used for human too. Here we are specifically concerned with the animal. Okay. So, now there are different modes of delivering these nanomaterials. Either you deliver them direct, or you deliver them as a carrier, or you deliver them as entrapped, something like a capsule like delivery. Okay. And there are a lot of polymers, polymeric nanomaterials, polymer coating, polymer gels and several such carrier modes have been used to deliver these particles. So, what we will do from here is, apart from it, we talked about the drug and nutrient delivery, which is antibiotic delivery to the targeted sites and site directed delivery of antibiotics or antibodies, then the biocides in terms of the metals, diagnostic tools using different kind of fluorescent probe, and there is a lot of work which is going on in terms of single particle tracking. Okay. Then you have a lot of reproductive aids, purification of sperm through the removal of damaged spermatozoa via surface marker recognized by nanoparticle. Similarly, there is another area which is exceptionally important, especially in modern animal production, is long term storage of sperm. Long term storage of, I should say, quality sperm, because these sperms are stored in cryopreserved chambers, and during this storage there are significant amount of damage which occurs. Now, how you can cryopreserve them, or there is another technique which is you are converting them into glassy structure, which is you are making them, you are shifting the cooling curve in such a way that ice spicules or crystals are not forming, which is called vitrification. So, how one can, but the whole idea is how to reduce the damage of stored sperm. So, this is very essential because whenever animal breedings are being done, if you find a good male whose sperm could be fairly effective to increase the production of the offspring it will generate, the common practice is that you preserve those sperms for posterity, and this is happening all over the world. You have amazingly good animal sperm banks where these are being preserved, but in order to preserve these kind of sperm, it undergoes a lot of damage. So, this is one area where there is tremendous amount of research goes on in places, especially in places like National Dairy Research Institute in India at Karnal, in Indian Veterinary Research Institute at Izatnagar, and several other veterinary and agriculture and dairy universities and industries across the country, in Gujarat and other places, where lot of efforts are being made to preserve these sperms in a way, and I will come to that, where all some of these nanomaterials could come very handy. Okay, then we talked about a little bit about molecular biology techniques, where nanoparticle based gene delivery, gene transfer and DNA transfection. So, these are the areas where a lot of concentration is happening. So, today I will take a case study of one of the nanoparticles, the way we did for the plants. So, we enumerate much of it, the wide range, but then we took one or two examples where we kind of highlighted the whole journey. The one which I am going to take up today is cerium oxide. So, cerium oxide nanoparticles or nanomaterials. Okay. So, cerium, those of you who are well versed with the periodic table will be knowing, or those who are not well versed will all remember that if you look at the periodic table carefully, so something like this, okay, and then you see there are two lines underneath like this. Okay. This all of you will remember, and one we used to call as lanthanide La series, the other one is actinide series. So, if you look at the lanthanide series, if you look at the second element, this reads as Ce, cerium, and please do not confuse with Cs, cesium. Many people does confuse it cesium. This is cerium. Okay, and much of these elements which are there, they stay or they survive at multiple oxidation states. The first one is lanthanum, then you have cerium. So, cerium is commonly used in the industry for multiple applications which has nothing to do with living systems. The area where cerium is used is in catalysis, in rocket fuels, in UV absorbing glass, so which is they find application in space research, in automobile research, and some of those heavy engineering areas. Now, just onset of, today we are into 2018, very early, like around 2000, I would say 2000, 2004, sometime around 2002, there was one idea which was tinkered back in University of Central Florida, Orlando, by one Indian individual, Professor Sudipto Seal, along with one of his collaborator, that Rzulinski. Could we use, it is used for catalysis, it is an antioxidant, it's an inorganic antioxidant. Could we use this as an antioxidant for biological systems? It was at that time this kind of crazy idea was taking shape. But how? So, initial studies showed something very unique about this material. So, when it goes in the nano domain, it does some very interesting reactions. So, I am going to first of all share the reactions with you, okay, how it acts. So, cerium in its nano domain, if you look at it. So, cerium, as I told you, if you look at this part of the game where I said you they remain in multiple oxidation state. So, let us start. What are the oxidation state of cerium? You see, so cerium remains in Ce3 plus and Ce4 plus. These are the oxidation state of cerium, okay, 3 and 4. And when we talk about a 10 nanometer or a 2 nanometer particle, we are talking about, if you typically calculate how many atoms will be there, keeping carbon radius in mind, there will be, I don't know, 5 to 10 nanometers. We are talking about something under 17, 18 or maybe 20 odd atoms are there. At that structure, at that geometry, it does some very interesting surface chemical reactions. It behaves like a autocatalytic molecule. As a matter of fact, this was one of the very first time autocatalytic ceria was proposed, or this word was coined by Professor James J. Hickman in University of Central Florida. In one of his discussion, he highlighted, oh, it seems it's an autocatalytic. Means, I will show you the reaction. What does that mean? It is changing its oxidation state all by itself, auto, okay, auto mode, okay. So, let us put the reaction in front of you, that will kind of make more sense. C3 plus, okay. Now, C3 plus does something very interesting. C3 plus has to get oxidized to become C4 plus. But if it has to get oxidized, essentially what it will have to do, it will have to give away an electron. If it gives away an electron, what it does in that process is C2O3, which is oxidation state 3 plus 2, you have OH free radical sitting there. Now, that OH free radical transform into CeO2, okay, balancing it. So, this is oxidation state 4 plus 4 of cerium, of course, okay, and here you have cerium in plus 3 state plus 4 state plus water H2O, okay. So, this action is essentially, so here you have the free radical sitting there. So, this free radical OH dot got an electron and become H2O. Now, this process is the free radical scavenging part of cerium. free radical scavenging aspects of ceria nanomaterial. Okay. Now, there is another set of surface reaction which happens, and that what provokes Professor Hickman to suggest the other way around it. That was, so if it is doing this free radical scavenging, let us highlight that part. So, H2O2 which is remaining in 2. So, this is how there is a free radical generation which happens in hydrogen peroxide. Free radical generation. Okay. Now, this C4 state of ceria reverts back, or it gets reduced. In that process, it will pick up an electron from the surface, and by picking up an electron, so this is the reducing electron and this is the electron which is helping the ceria to get oxidized. So, this electron which it picks up, it's basically a recovery by surface chemical reaction. This kind of recovery is called the recovery by surface chemical reaction. RX stands for the reaction. So, what is happening is to CO2, which is oxidation state 4. So, it has to be a slightly aqueous system. Okay. It forms Ce2O3 plus half O2. It is generating oxygen there. So, you realize, and this whole process, the recovery by surface chemical reaction. So, this whole thing, what I have put across here, this whole reaction is in a auto mode, or this is what Hickman seal highlighted upon, that this is autocatalytic system. Okay. And specifically, this autocatalytic process has its own tremendous implications. What are the implications? We will talk about it now. So, this process, what you are observing, leads us to a situation where you have a finite amount of this material, and it is just, what we are talking about, 3 or 4 atoms to ceria, couple of oxygen hanging out, dangling out there. This gives us a situation where a very finite amount of material could continuously auto-regenerate itself and results in performing antioxidant activity. In other words, what we are talking about is it has a pseudo infinite half-life. In other words, because pseudo infinite half-life So, we will continue this discussion from here. What are the physiological implications in animal body for a material which has a pseudo infinite half life? Thank you.